to now ask Ulrike Ziehm to give her presentation. She is Senior Biomedical Data Scientist at Molecular Health GmbH in Heidelberg. She did her PhD at the Humboldt University in Berlin, followed by a certain time at the Ludwig Maximilian University in München, and is since 5 years with Molecular Health. So I hope I can see she is on, but has not unmuted and is not, yeah, now it's gonna be unmuted, and I hope she's sharing her content with us. In a second, it's uploading. And there we are. And Ulrike, it's your floor, and you can start giving your talk. Thanks a lot.
Yeah, thank you for having me. Everything's said already about me. I'm data scientist at Molecular Health, and I will present today a study where we were comparing the performance and robustness of two TCGA-inspired risk stratification strategies for endometrial cancer on a single DNA sequencing platform. Very short introduction to our software, MHGuide. MHGuide is a software that enables evidence-based clinical interpretation of NGS analysis. In this case, we're talking about tumor NGS data. Guide supports the interpretation of sequencing data from all kinds of platforms and gene panels, including whole exome sequencing, and uses a comprehensive database of quality and expert curated variant interpretations. We call this clinical variant interpretations. And in the end, it will provide a report on the analyzed tumor DNA sample, where therapy options are provided. So let's say potentially effective treatments, also information about potential risk for resistance and safety issues for certain treatments, as well as information about clinical trials. So in a bit more detail, the workflow. Starting from a tumor DNA sample, we can start from FastQ, BAM, or VCF, whole exome sequencing or panel of choice. If we start from FastQ, MHGuide has the full bioinformatics pipeline from alignment to variant calling integrated. So it's calling then SNPs, indels, copy number alterations, MSI status, and also gives the tumor mutational burden. And via VCF input, we can also interpret gene expression data, protein expression data, methylation, MNPs, and so on. The biomarkers are categorized by our internal CVI score into by basically the development status, so preclinical, clinical, clinically approved, and also the AMP score, so pathogenic, likely pathogenic, these kind of things. And treatments are annotated with their also their development status. If they are approved in the specific cancer of the patient, or if they would be off-label, or if they are still developmental drugs and so on. Clinical trials are given, as well as medical guidelines. And in the end, you will get a report which lists the potentially effective, ineffective, and potentially safety risk treatments, as well as prognostic and diagnostic biomarkers. So this is the technology on which we were basically doing this, this study, starting from FastQ. I will go into this in detail. Endometrial cancer has been or is classified classically by histology into type 1 and type 2. Type 1 is endometrioid, estrogen dependent with clear precursor lesions and has typically a good prognosis. Type 2 tumors are other histologies, serous, clear cell, and so on. They are not hormone dependent. There's not such clear precursor lesions, and they have typically a poor prognosis. They are generally regarded as high-grade tumors. And these types are also characterized by specific molecular characteristics, specific mutations, and molecular markers have increasingly been evaluated in clinical practice to add, for example, aid reliability of histopathological diagnosis, to improve risk stratification, and to guide treatment decisions like who could benefit from a chemotherapy and who does not benefit, things like these. Quite a while ago already, in 2013, the Cancer Genome Atlas Consortium, TCGA, did a comprehensive molecular profiling of endometrial tumors on, I think it's 6 different platforms. So they did whole exome sequencing, mRNA expression, protein expression, microRNA expression, methylation, and microsatellite PCR on these tumors. And they found 4 integrative, that is multi-platform defined subtypes that have clear prognostic indications. One of them, called POL, is characterized by super high mutational burden, specific mutational pattern, and, well, as the name says, mutations in POL-E, and has, irrespective of grade and stage, excellent outcome. So I want to point here to the observation time. This is around 10 years follow-up time here. And there's one class that's called copy number high. It's characterized by, well, as the name says, high copy number counts. That is the poor outcome class for endometrial cancer. So, and the other 2 classes, MSI and copy number low, are like intermediate risk for endometrial cancer. The MSI class is also characterized by high mutational loads, not as high as the POL class, but also high, and characterized by microsatellite instability. So they might have actually implications for treatments. Those 4 subtypes can actually be classified on 3 platforms only, not all 6 are required. The POL class is identified based on the SNF counts and the specific nucleotide substitution pattern, the C to A and C to G transversion rates. Notably, to classify the POL cases, the POL mutations are not actually taken into account. It is just it's just classified based on the SNF counts and the nucleotide substitution rates. The remaining tumors that are not POL are identified as MSI class if they have high microsatellite instability as detected by MSI PCR. And the then still remaining tumors are classified as copy number high if they fall into the copy number cluster 4. So that's based on microarray data, which has been clustered before. And the remaining cases basically go into the copy number low class. And since this is quite unfeasible in clinical practice to this day to do this comprehensive profiling, there have been early on efforts to find, I call it surrogate markers of these subtypes to do the classification with less effort, with less tests. And I just give 2 examples here. One is Talhaug et al. Study from 2015. And there's a series of studies called PORTEC, still ongoing, some of them that also use these surrogate markers to classify patients into at least TCGA-inspired subtypes. The typical markers used are mutations in POL as a surrogate for the POL class, MSI status, or mismatch repair deficiency. So this is used kind of synonymously. So either detection from MSI PCR of mismatch repair gene expression. And for the as surrogate for the CN high class, they use p53 expression or mutations in TP53. And some use additional markers, but I will not go into this here. So the platforms may differ, the exact scheme of classification may differ, but yeah, the common is they all use these surrogate markers. So the objective of this study was, can we reproduce this three-platform TCGA rule, this original rule which uses the SNP counts, the MSI status, and the copy number cluster to classify the patients? Can we use this rule on parameters derived from DNA sequencing only using MHGuide? And does the surrogate marker classification work also from DNA data from MHGuide? How do they perform with respect to classification, performance, and risk stratification? How do they compare? And how robust are these two classification methods with respect to generalization to other data sets? So we evaluate this on TCGA data, but also on an independent data set. Yeah, there we go. So little overview of what we've been doing. We analyzed 232 endometrial cancer cases that were classified into these 4 classes from TCGA in paired whole exome mode. And we started from FastQ. So we downloaded the BAM file from TCGA and extracted the reads and ran our full pipeline from alignment on. Implemented this classification rule from TCGA using the classification parameters derived from our MHGuide analysis, and implemented the classification by surrogate markers also as derived from MHGuide analysis. And we will look into at the classification performance, how good do they reproduce the original TCGA classes, and the risk stratification, how good do they actually split the patients into high and low risk. And we had an additional cohort of endometrial cancer cases from the University of Greifswald, also paired whole exome, also analyzed from FastQ. They were FFPE samples, in contrast to the fresh frozen samples from TCGA. And they had comparable follow-up time as the TCGA study. So we have up to 10, 12 years follow-up time for these cases and extensive clinical data, treatment, and even adverse reactions. Yeah, so starting with the classification by this TCGA rule. So this was the rule I've already explained. We replaced the the SNPs we replaced with the SNPs called by MHGuide. The MSI PCR results we replaced with the MSI status that we call within MHGuide from DNA NGS, and also the copy numbers we call within MHGuide, and we trained a supervised classifier, a naive-based classifier actually, to the copy number 4 cluster of TCGA to predict this cluster basically. And using these parameters, we try to reproduce the classification. So just this is maybe what you have to remember most through the talk. The POL class is the lowest risk class. This copy number high class is the high risk class. I will mostly focus on those 2 classes. The surrogate marker classification, we decided to use a very, very similar classification scheme. So classifying the patients in the same order as TCGA did. but instead of using SNP counts and nucleotide substitution pattern, we use pathogenic or likely pathogenic pole mutations to identify the pole cases. Still use the MSI status that we get from MHGuide to classify the MSI cases, and use the somatic non-silent TP53 mutations to identify CN high cases. Looking at the classification performance here, this is the original distribution of classes in TCGA. And here on the right side, now you see the distribution that we get from our two different stratification schemes or classifiers, let's say, which looks very, very similar. Of course, we could have them totally wrongly classified and still get the same distribution. So looking at the overall accuracy here, we get pretty good results. So left-hand side, the TCGA rule classification from DNA and GS data, right-hand side, the surrogate marker classification, both reach around 90% accuracy, precision and recall. Looking into how good we are actually at identifying the good outcome class and the bad outcome class. So for the identification of the pole class, the TCGA rule performs perfectly here, identifying all of the pole cases correctly, while the surrogate marker-based classification is a little less sensitive, so recall is a bit lower. It also has, I think, one false positive, but it's still really, really good. For the CN high class, we are not that perfect, but still very good with an accuracy of around 90%. And here again, what's seen is that the surrogate marker-based classification is a little less sensitive and a little more precise than the TCGA rule. So it identifies fewer people as CN, or fewer tumors as CN high. But those it identifies are very precise. Now, when we look at the risk stratification, looking at the Kaplan-Meier plots on the, that's the left side right here, yeah, left side, the original subtypes, right side, again, our two classification schemes, and this looks very, very similar. I think we can all agree. Also looking at the hazard ratios, we measured the hazard ratio versus the CN low class for each of the classes. So as the CN low class is kind of the rest of them all, we use this as the reference class. That's debatable probably, but this is how we did it. So for all three, so for all our classifiers, we get this very low hazard ratio for pole, this around 1-ish hazard ratio for MSI, so it's very similar to the CN low class. And for the CN high class, we get a hazard ratio of around 3, let's say, roughly. Summarizing this part, on this TCGA cohort, both of our classification schemes using only DNA and GS data perform pretty good and very comparable with respect to classification and risk stratification. So yeah, now we look at our independent cohort from the University of Greifswald. Of course, there we don't have a multi-platform classification. We don't have ground truths here, so we can only look at the risk stratification to see how good the two classification rules perform. This is the distributions that we get for the two rules, the two classifiers. Similar to the TCGA cohort, what we see is that the surrogate marker-based classification puts a little fewer tumors into the extreme classes, so a little less into the pole, a little less into the CN high class. Now looking at the actual risk stratification performance, that is the TCGA rule. And you can maybe imagine we were not too happy about this. So it just didn't perform. There's nothing to deny here. However, the surrogate marker-based classification still performed pretty good, apparently. So Kaplan-Meier curves are great to lie with. So we look at the hazard ratios. They actually confirm this. So the TCGA rule just completely not performs, in particular, not to identify the high-risk patients, while the surrogate marker-based classification identifies low-risk cases and high-risk cases very, very well. Questions now, why is that? So one of the things that always happen between cohorts is that you have data shift, you have distribution shift, and in principle, one could adjust for batch effects. However, in clinic, so in our day-to-day practice, we usually don't get batch data. So, and also I sorry, sort of doubt that this would have helped here. So the problem is with the TCGA rule relies on MSI and CNA calling from NGS data, and both show to be quite sensitive to the quality of samples. And our independent cohort, unfortunately, sample quality-wise was not that not as good as we would have wished for, but on the other hand, it showed us, for example, very strong systematic dependency between the MSI calling and the fraction of short fragments and duplicates. So we learned a lot from it. Yeah, but yeah, those are the reasons for the performance differences on the two cohorts. So summarizing and coming back to the objectives, the feasibility. So in principle, yes, we can actually even do the 3-platform TCGA rule on parameters derived just from DNA NGS data, given good data quality. And yes, the surrogate marker classification works also from DNA NGS data. And on high quality data on the TCGA data set, both classification schemes worked comparably good and actually pretty good in classifying the patients. And I think most important takeaway is that only the surrogate marker-based classification generalizes to lower quality data. And actually, I think it's pretty nice to see that it is robust really to a wide range of data qualities and still performs very well on this independent data set. Yeah, so I think that was that. I was fast. I want to thank the organizers of this meeting, and in particular, our thanks goes to Alexander Muster and his team from the then University of Greifswald. He's in Bonn now, and he's the one who's been providing us with the data of the independent cohort. And also thanks to the team at MH that was involved here, in particular Rahel Hartmann, who was actually doing most of the all-hand work, of the hands-on work on this project.
Thank you, Ulrike, for your interesting talk and for the insights you gave us. My question is, how can the quality control be standardized across different laboratories according to the needs? So you have seen that and your assumption is that the quality of the samples have a large effect on the analysis of the data and the stratification afterwards. So how can we assured that the quality is similar?
So MHGuide provides with a quality report as well for the whole sample. So we did actually do a very thorough analysis of the quality parameters, like the fraction short fragments, fraction of duplicates, adapter contamination. We looked, of course, for the for signatures of FFPE artifacts and so on, and correlated this with, for example, the MSI calling scores that we received. If you get a batch, you can do that, and that's nice. And you could even correct for it, or you can just say there's nothing to be done. So some things just don't work if your DNA input is too degraded. So for everything, so what we did. So of course after this, I had quite some meetings with the QA department. So we do have warning messages if certain quality parameters. So generally, if quality parameters are above thresholds, MHGuide will tell you. It will show this as a warning signal for the whole case. And also, I think on a variant and biomarker basis, it gives well, variant level quality information. So in this case, we decided that we would probably not show anything that is based on MSI calling or CNA calling on samples that have very bad quality. So fortunately, I think this is not the average quality of data we get. But on the other hand, I think it is very good to have data of a wide variety of quality to, well, just get an idea of where's the limit on which you can still make, reliable statements on, certain biomarkers.
We have heard in earlier presentations that they rely on a similar question on the DNA integrity number, and with RNA on the RNA integrity number, the RIN number. Would you suggest a similar approach to look at those numbers? And if yes, what numbers do you consider as being okay, and which ones are not okay?
This I cannot answer to. I'm not a bioinformatician, so I'm not involved actually in the pipeline here. I'm a data scientist. But yeah, I was very interested in that talk, and I will definitely check into this and suggest maybe including this into our quality analysis.
Okay, then thanks a lot. So we are